DNA replication. Then how, in summary of this, we will discuss it. Replication is always semi-conservative. It is a first rule. Replication begins at the point origin of the replication, start, or we can say the start point. DNA synthesis initiated by the short fragments, you know, because it is initiated in uh, in eukaryotes, it is 100 to 200 nucleotide long. In prokaryotes, it is 1000 to 2000 long nucleotides long. Elongation of DNA strand always in the 5 prime to 3 prime direction. So new DNA is synthesized from the DNA TPs, and for synthesis of the new strand, growing strand requires the short RNA fragments, that is a primer, that is known as the primer. Okay, and hence the polymerization of the DNA molecule takes place by addition of the deoxynucleotide triphosphates, and in from this triphosphate, the diphosphate, two phosphate groups are cleaved from the deoxynucleotide triphosphate, and then resulting nucleotide is the 3 prime OH group of the growing nucleotide strand. So the replication is continuous. Leading strand, it is synthesized continuously, and discontinuously. Lagging strand, it is synthesized discontinuously. That I already told you. The new nucleotide strand are complementary and anti-parallel to each other. Okay. So replication takes place at very high rate. Uh, Generally, the rate of this enzyme, if I said that the DNA polymerase third enzyme adds the 1000 nucleotide per second, it is a very fast rate of the DNA synthesis. So the replication of the DNA synthesis was first accomplished by the scientist Arthur Kornberg and his co-worker in 1950. Kornberg received the Nobel Prize in 1959 because this scientist isolated the enzyme which is responsible the synthesis of the DNA. Hence, this enzyme is also known as the Kornberg enzyme. So, called as the Kornberg enzyme. Okay, and this enzyme was the DNA polymerase first enzyme. So there are three kinds of the DNA polymerase enzyme nowadays identified: DNA polymerase first, DNA polymerase second, and DNA polymerase third. We are going to discuss more about these enzymes. But what are the requirements of the DNA synthesis? Okay, that we are going to discuss. So for the DNA synthesis, will require the primer. DNA polymerase first or second, not first or third enzyme cannot initiate the synthesis of the DNA molecule or DNA strand. It requires the primer. So this polymerase third enzyme. So this enzyme requires the primer. The second is the template. The parental strand. Parental template strand is required for the synthesis of the DNA molecule. So then we are going to discuss this process. Again, the proteins, which are the other proteins required for the DNA synthesis? These proteins are nothing but the DNA helicases, then single strand binding protein, then topoisomerases, and DNA summarized all these things here. So the primase enzyme which adds the primer. Okay, then DNA polymerase first, DNA polymerase third is required for the synthesis of the DNA molecule. Then RNase is also required for primer degradation. Then the sliding clamp, it is a part of DNA polymerase enzyme. That is the beta subunit of the DNA polymerase enzyme, and then DNA ligase enzyme. So DNA helicases enzyme. What is the function of DNA helicases enzyme? This enzyme binds to the double-stranded DNA and breaks the hydrogen bonds and separates the two strands of the DNA molecule. Okay, like this. separates the two strands. So separation of the both the strands of the DNA, it is done by the this DNA helicases enzyme. Then DNA single stranded binding protein, SSB protein, these are known as SSB protein. Then these proteins binds to the single stranded DNA molecule and stabilizes it. So two functions of this single strand binding protein are, the first function is it protects the single stranded DNA molecule. Otherwise, whenever there is a formation of single stranded DNA into the cell, it is degraded by the nucleases enzyme. It is attacked by the nucleases enzyme. So to protect that DNA, this DNA is bounded by the single strand binding protein. Then after that, the second function of this single strand binding protein is it prevents the reformation of the double helical structure of the DNA molecule. Okay, so this is the important. Then the DNA gyrase enzyme also catalyzes the conversion of the positive supercoil form of the DNA into negative supercoil form, because into the cell it in DNA it is very long molecule, and to accommodate very small cell, it is wounded in very specific manner, and it is packed into the very small bundle, and it is packed into the cell, so that that compact wounding, it is called as the positive supercoiling of the DNA molecule. And for the replication of the DNA molecule, there is a requirement, it should be in its negative supercoil form. Then conversion of the positive supercoil form of the DNA into the negative supercoil form, it is done by the DNA gyrase enzyme. Primase enzyme, what is the function of primase enzyme? They adds the primer at 3' OH group of the template strand. So primer is added by this primase enzyme, and then DNA polymerase third enzyme comes here and binds to the primer. And when it binds, that complex, it is known as a primosome, like this. Okay. Then next, DNA polymerase enzyme. Here, the two kinds of DNA polymerase enzymes are important: DNA polymerase first and DNA polymerase third. DNA polymerase third enzyme, it is considered as the real replicating enzyme, and DNA polymerase first important for the synthesis of the lagging strand of the DNA molecule. Okay, so I am going to show you in the figure in next slide. So RNases, what is the work of the RNases? To degrade the RNA primers. These RNases are required. DNA ligase, to join the DNA strand, this DNA molecule requires, because they forms the phosphodiester bonds into the during the DNA synthesis. So this is the theta model of the DNA replication. So at the point origin of the replication is there, means this is the rule, one of the rule of the DNA replication, where the DNA replication takes starts from at one point. Okay, so this one point, there is a separation of two strands are there. Okay, and this separation of the two strands, replication bubble is formed. This is the another rule. So replication bubble or replication eye, it is formed. And here the two replication forks are moving. One is moving in this direction, okay, in right hand side, and another is moving left hand side. And in this way, by moving this replication fork, they comes at one point, they comes and here they meets at specific point, that is we can call DNA termination site. DNA replication, termination of the DNA replication takes place, and from the one double helical DNA molecule, two daughter DNA molecules are synthesized. Two double helical daughter DNA molecules are synthesized. In this DNA replication, initiation of the DNA replication starts at the specific point. That specific point, that is known as the initiation or origin of the replication. that is called as the origin of replication. At this particular point, the DNA replication starts. And by binding with the DNA, A protein involves in this here initiation of the DNA replication. And by binding of the DNA A protein, the two strands of the DNA molecule are separated by breaking the hydrogen bonds. Here, these hydrogen bonds are break by itself. Here, the DNA A protein and the initiation of the DNA replication starts. And here the helicases enzymes are get bounded to the both the strands of the DNA molecule, and which separates the further DNA, both the parental DNA strands. Now in this figure you can see here the this is the blue, dark blue color. This is it is the parental DNA molecule. You can see here, this is the first protein, that is the topoisomerase. I already told you the uh, function of this protein, that is the conversion of the positive supercoiled form of the DNA molecule into the negative supercoiled form. So this DNA molecule will ready for the DNA replication. And here the next enzyme, this is the helicases enzyme. You can see here in this figure. This helicases enzyme, what is the function? What the hydrogen bonds are formed in between the base pairs. Adenine pairs with the thymine by forming two hydrogen bonds. Guanine pairs with the cytosine by forming three hydrogen bonds, as we discussed in structure of the double helical DNA molecule. And separation of these two parental strands is done by the helicases enzyme. And here's the after separation, when DNA molecule comes in the single strand form, then it is bounded by the hexameric protein, that is known as the single strand binding protein. Single strand binding protein maintains this DNA molecule into the single strand form. Okay, it prevents again formation of the double helix of the DNA molecule. And second thing, it protects from the nucleases enzymes. Now two strands are separated here, and DNA replication then starts. Then primase enzyme, first important enzyme, comes here and plays the role of this enzyme. That is, it adds the small primer, that is the small segment of the RNA in particular direction. So this is the direction. At the 3' end of the template strand, it adds the primer, small primer. And then new strand, because the parental strand is having the direction of the 3', then this new strand will have the direction of the 5, because these are anti-parallel to each other. So the 5' strand, daughter strand, it is synthesized. This is red color is the primer. Okay, and to the primer DNA polymerase enzyme is bounded. This DNA polymerase third enzyme, it is you can see third enzyme, and one strand it is now synthesizes the one strand. Okay, so other side, you can see here the DNA replication, it is going on. This is a light blue color strand, you can see. This is the daughter strand, and this is the parental strand, dark blue color. And this light blue color strand, it is continuously going to synthesize, continuously synthesizing this new strand. You can see it is a continuous one. So this strand, it is known as the leading strand. Okay, and this all parental strand, it is known as means it is served for the synthesis of the leading strand, hence this template strand. And while here it is synthesized into the short segments. After that, further it is a discontinuous, it will stop here, then next will synthesize, and then it will stop here, then next it will synthesize. In this way, this DNA replication takes place, and synthesis of the both the strands takes place. Continuously synthesizing strand, it is known as the leading strand, and discontinuously synthesized strand, it is known as the lagging strand. And the synthesis of the lagging strand was discovered by, or was extensively studied by the scientist Rizi Okazaki, and hence these fragments are known as the Okazaki fragments. Okay, so how this synthesis takes place, that we are going to discuss in next slides. So this is the helicases enzyme. You can see here, this is the helicases enzyme which is separating the two strands of the DNA molecule. And here the another strand, it is a continuously synthesizing strand. This is the green color, it is a primer. It is added at the 3' OH end of the parental template strand. And then at this point, DNA polymerase third enzyme is binds, and after binding, it moves in this direction 5' to 3', and it is moving here, and it is synthesizing this strand. So this strand, it is continuously synthesizing, hence we can call it is the leading strand. But you can see here this strand, the small RNA primers are added, and small segments of the strands are synthesized, small fragments. Maybe in case of prokaryotes, 1000 to 2000 base pair long. And these fragments are known as the Okazaki fragments. Okay. So these Okazaki fragments, then later on, the RNase H enzyme comes and breaks these primers, and further DNA polymerase first enzyme adds, bounds here and adds this um, or fills these gaps which are created by the primer degradation. So you can see here in this figure,